Yeah, Germany, Finland, hello. Okay. Yeah, Saudi Arabia, Madrid, Portugal. And we have also people from different time zones. So for most of us, it's it is 1:30, but there are people which they are early in the morning, and other people which are late in the evening, like Martin. And thank you for joining this tutorial, even if it's not the best time for you. So I think we can smoothly start. We have 90 participants, and yeah, welcome to everyone. We are looking forward to give you this introduction into metagenomics. As you have seen in the description, there are actually two groups, two tutorials joined together. One is using metagenome-atlas. It's mainly developed by me and Sofie helping me to answer it. She's really an expert in this tool. And the other group is from Martin Steinegger and Milot, which have developed this MMSEC family. You want to say hello?
Hi, everybody. Looking forward to explain more about MM6 later and to answer your questions in the chat.
Hey, everyone.
Hello, thank you. So the structure of today, we have, no, first, if you have questions, ask them ideally by this live Q&A. We are trying to answer it during the tutorial or in the breaks. We have also put this web page where we have an access to a chat. And if you have bugs or problems you cannot install it, put it there. And this chat room will actually also be available after the tutorial. So if you want to try this tutorial probably this week, we know that we cannot do everything in these 3 hours. If you have time later on, you can ask questions on this other chat room. But for today, please, if you have questions, try to answer them, to ask them in the live Q&A. So for today, we have this first part of metagenome-atlas. I will give an introduction to metagenomics in general, then more details on the atlas tool. We'll have a short break, and then we start with the tutorial. And in the second part, I hand it over to my colleagues, and they will give an introduction to the MMSEC tool. Again, try to at least click on the web page with all the links. So I think I can start. Introduction to metagenomics. Metagenomics is really the genomics of a whole microbial community. Instead of culturing one microbe one by one, we are studying them in isolation. We are really trying to sequence them and to sequence them in their natural habitat. And this can give us really insight in how the microbes are in their environment and analyze functions and the composition. From a wet lab side, the metagenomics more or less consists of taking a sample. You take the sample from either water, from the gut, or from soil, or from mollusks, any environment where you can extract a sample, a DNA. You extract then the DNA, and this you send for sequencing. Even though more and more long-read sequencers are becoming popular and more used, most of sequencing today is still done in Illumina. And for this, we have to shred the long reads, the long sequences, because Illumina is more adapted for the short reads. And due to this shredding, we call it shotgun metagenomics. The output of such a sequencer is then usually handed over to a bioinformatician, which mainly consists in files with the short sequences. And then we are told, okay, here are the sequencing results, analyze the data. And today we want to really give you an introduction of how you can do with this sequencing data. So the first, probably the simplest, is to map it to a database. You map your short sequences to a database, and this can give you really the idea of based on what is known, but usually this can only be a small fraction of what is really there. Another approach which I find more interesting is try to put this short sequences together and try to assemble genomes which are may also contain the genomes which are unknown and are more specific to your sample, with the goal that you can recover much more of the reads or in your data. I just stopped this one because it's kind of annoying. And then okay, next step is so how we can, so the goal is how to get genomes out of the metagenomic sample. And for this we use assembly. In theory, very brief, the most assembler use what is called an algorithm based on the De Bruijn graph. You cut your short read into even shorter sequences of fixed lengths, the so-called k-mers. Here an example for k equal 4. So you cut this sequencing read into pieces of length 4, and you make a graph of it with the k-mers representing the nodes and the read representing a path in this graph. Obviously, this is the theory, and in practice, it looks much more complicated. Here is a graph of an assembly from a metagenome of a gut. And yeah, it's much more complicated because we sequence a whole community of microbes. So the genomes are overlapping. The reads are not covering the genomes uniformly as it would be in a single genome sequencing, but it's much more variable and usually lower coverage. And another problem is that a lot of these genomes have regions which are highly similar. For example, the ribosome is conserved almost in all the phyla, in all the domains. And in this example, we see that there are two regions with highly connected clusters, and they represent the two most dominant phyla in the gut, the Bacteroidetes and the Firmicutes. And the edges between probably represent these regions of the genome which are conserved even between the two phyla. So unfortunately, it's rarely possible to really assemble a whole genome from the metagenomics data. And you end up with a lot of longer sequences, the so-called contigs. But you still can do a lot of them. You can, for example, try to annotate genes. And even there are ways to annotate the taxonomy to these longer sequences. You can then quantify them and try to compare them between different samples. And this is really what was done probably in the earlier days of metagenomics, trying to look for genes, gene catalogs. And it probably still is done today when you cannot assemble a metagenome. On the other hand, in recent years, more algorithms were developed where you can which try to group these contigs together, and to group the contigs together which probably come from the same genome. We call them metagenome assembled genomes, or MAGs. And yeah, using really these algorithms of binning, as they're called, many publications are able to assemble thousands of new species which were not known before, which were not cultured. And yeah, studying really new environments. On the other hand, these MAGs are not perfect. They are usually fragmented. They are not complete. And the spinning algorithms may also group contigs together, which are which from different strains or even from different species. So we need a way to assemble the quality of these MAGs. Of course, if you have already a reference genome, then it's kind of easy. You can compare the MAG to the reference and see how complete it is. But if it's an unknown species, how do you want to assess the quality? And the solution is to look for marker genes. Oh, yeah, I forgot that. So here, marker genes. So the marker genes are genes which should be in each genome or in each genome of a family, or are present at 90% of all the genomes in a family. So they are really essential genes, core genes. And you count more or less how much of these core genes are present to what is expected for this family, and these are then your completeness estimation. On the other hand, you look for genes which are usually single copy in this family, and you say that is if you have multiple copies of this single copy gene, then it's probably a contamination. back here, binning usually can use different features of the context. You can look at the sequence composition, like the tetranucleotide frequencies, which kind of captures the or embeds the idea of GC content or the codon usage. So this is one of the measures used to group context together. And of course, the other is the abundance. If a genome is high abundant in a sample, all the parts or all the context of a genome should be high abundant. If it's low abundant, all the parts of a context of a genome should be lower abundant. You can also combine different samples and look how abundant it is in different samples, so difference in abundance between samples. All this can be used to group context together and get this max. You can also combine different samples for assembly, the so-called co-assembly. But usually this makes things even more complicated. So if it's complicated enough in one sample, it's even more complicated if you put several metagenomes together. But the advantage is you can maybe recover genomes which are lower in abundance. But it's difficult to scale as well. Then if you use the single sample assembly, usually you can actually recover the same genome in different samples. So the same species or the same genome in different samples. And ideally, you want kind of de-replicate this redundancy or group these genomes together. And we do this usually by comparing all the genomes in a pairwise. We calculate the average nucleotide identity, the similarity between the genomes. And here I put a graph of, I think, 5,000 genomes. And we see that many of these dots, which represent these genomes, cluster very well together. So they represent probably the same strain, or at least the same species. Today, what we do is we take a cutoff of 95 ANI as a species threshold, and usually group the max with this threshold. So this is great. That's the way how we get the genomes out of metagenomes. But what is next? So what is next? Ideally, if you have genomes, you want to annotate them with taxonomy and functions, and also quantify the genomes for at the end be able to compare it and make some new statistics. Yeah, make some new paper out of it. In the next section, I want to discuss the metagenome-atlas pipeline, which actually does all of them. The metagenome-atlas pipeline is a pipeline, really doing implementing all the steps from quality control assembly, the genome binning, and the annotation. And so I will give more details how I implement these concepts in metagenome-atlas. But first, some publicity. I really worked hard to make it easy to install, easy to use. And the idea is you can start, if you have the conda installed, you should be able to start with 3 commands. So probably everybody, even non-bioinformatician, can do it. And of course, from time to time, I get reports from users, which it's not so simple to install, but it works. I helped somebody to install it for his company. And once they have the conda installed, they could start in 3 commands. So that's the goal. And as already told, the metagenome, especially the assembly, takes a lot of computational resources, a lot of memory threads, and you need to apply metagenome-atlas and this assembly on a cluster or a cloud system, which adds some more complication to make it work. But I made some wrappers for the common cluster systems, and thanks to the underlying snake-make, the Atlas pipeline can actually submit jobs to your cluster. This looks like this. So you have one job, for example, the get read stats step in the Atlas pipeline, and it submits automatically to the cluster. Once it's finished, it starts a new job, and you can do different samples in parallel. So you can really use the full capacity of the cluster. And sometimes you have even to pay attention not to spam your colleagues. As said, I made some wrappers for LSF, the PBS, and Slurm cluster, but it can really adapt it to any cluster and cloud system. So a bit more in details about the steps of Atlas. If you start with raw reads, you have to do quality control, which really consists in remove the low quality bases, and you can also remove contaminant of common contaminant. So you have to remove contamination, which come from the library preparation or from the sequencer itself. If you take a microbiome or a metagenome which is host associated, you also should provide the genome of the host to remove the reads which are coming from the host rather than the microbiome. And yeah, the idea is to get good quality reads at the end. Atlas produces several an interactive report which can really help you to guide where are the reads going, in which step they are maybe lost. And here, for example, you see that in one sample, the quality filtering removes most of the reads. This means probably most of them are mapping to the host instead of the microbiome. So this helps you to assess the problems and maybe remove this sample. It gives also information about the read quality and yeah, quality report. If everything is fine, then you don't have to pay too much time on this quality report, but I think it's very useful. You can also start directly with quality, like already quality controlled reads and feed this into Atlas. The next step is assembly. I implemented Metaspades or Megahits, which are the two most popular metagenome assembler today. Metaspades especially is shown to be really good to recover marks at the end. The problem is only that it takes a lot of resources, it takes a lot of memory. And even in big groups, you see sometimes they started with spades and then there's one sample which they could not assemble, even with 1 terabyte of memory. On my cluster, I don't have 1 terabyte of memory, so it was a bit tricky to still use it. But what I did is to implement the step which is takes the most memory, the error correction, outside of spades using a tool which is a bit more efficient. So we correct the reads to improve the assembly. And another step we can do is to merge paired end reads. If you have paired end reads, then you can merge them. And this works even if the paired end reads are not overlapping. So you can do a kind of pre-assembly and connect the two paired end reads, which then should improve the assembly later on. It's possible to use long reads in combination with short reads, so the so-called hybrid assembly for the metagenomic assembly. And yeah, at the end is you get the scaffolds or the context out of it for the last. Then in the we get the assembly report as well. You can see how much the length and the number of contexts to see how good each sample performed. Then for the binning, there are a lot of binning algorithms, and sometimes it's difficult to choose. And the good idea is you don't need to choose a binning algorithms. There's a cool tool called DAS tool, which can combine different outputs of from binners. So in Atlas, we use MetaBat2 and MaxBin2, which are also, again, popular binning algorithms. Then they're combined with the DAS tool, and quality assessment is done by Checkm. And you can use co-binning, so mapping the reads from different samples to the same, and dereplication to get really unique genomes for each species. They are then used, nicely named MAG1, MAG2, MAG3, MAG100, or whatever. And yeah, so you get the genomes out of it. Here is a plot of the quality from the test data actually used in the tutorial. So you see these dots representing again genomes with quality and the contamination, and you see that most of the marks are on the top left corner, representing high completeness and low contamination. Then again, for quantification, I use BBMap, which is fast. I calculate something like the median coverage over a genome, which is handy for calculating the relative abundance, because it implicitly normalized to the genome size. But you also can get the row counts, which then can help you do all the statistics with the row counts. So quantification. Then for annotation, probably used for, from your experience that the taxonomy is for prokaryotes is very complicated, let's say. This is still because one hand we don't have a lot of the bacteria in culture. On the other hand, kind of people started classifying them by phenotype under the microscope, and this doesn't have to be what is really the genome. A group from Australia made a new taxonomy, which is more based on the genomes. They also include the max, and so they really tried to make a taxonomy which is based on the differences in of the genomes, but still make some compromise with the traditional names. They have to split sometimes different taxa, which contains different lineages, and make it more robust. Here are some examples from their taxonomy. So the NCBI taxonomy, the order Clostridiales was split into many different orders because the genome showed that actually they are much more different than what the other orders are. On the other hand, for the Bacteroidetes order, class here, they are actually combining them together because the previous taxonomy was much too broad and than what is defined by usual genome distances. So I use this GDB taxonomy in Atlas to get something. It's also interesting because as they include max, you get maybe a taxonomic annotation of species 175, but it represents a MAC which was already found earlier. So you may not give a nice name out of it, but it still gives you an information that there is another genome from the same species. So this is for taxonomy. For the functional annotation, I use not only the genomes, but also all the contigs which could not been together into genomes. Then we predict genes on them, and we annotate them with EggNote Mapper, which is one of famous tool. Here again, if you have the same species in different samples, you end up probably with the same genes, and you don't want to annotate them the same gene several times. So I cluster these genes into a gene catalog, and for this actually I use tools from the MMSEC suite, but I let my colleagues explain that in more detail. So at the end, we end up with annotations. The EggNote can be linked to Keck, CASI, Cox, and much more. And here is some histogram output of the relative abundance of Kerk orthologs in different samples. It's one of the outputs you can get out of Atlas. So I think this is kind of what I have to say for Atlas. If you have questions, just ask them in the live Q&A. And then it's now 2 o'clock, half an hour of speaking. I let you start with the tutorial. The tutorial is based, you have 2 steps. The first, it's kind of analyzing the output of metagenome-atlas. And then in the second part, you can run the 3 commands on a dummy data set. Here's again, probably better if you have strictly, if you can work on your cluster, it would be the best. The tutorial is based on how it's, the question in the tutorial is how to analyze a metagenome from mice, which are fed high-fat diets, and to see what makes this obese mice, what are changes in the microbiome. But I let you click on the links, which is also on the landing page. If you have questions, then ask them. I try to answer them in a short break. And then probably at 1:30, I give the words to Martin and Milot for the MSEC tutorial. And if there's more time at the end, I can still finish the Atlas tutorial. And I'm here for any questions if you have something. Thank you very much. So there are many questions. I try to answer them in live. So Seja is asking, can metagenome-atlas also used for other than for bacteria and archaea? So can it, for example, be used for viruses? So the metagenome-atlas, the basic implementation is really for binning, and with that you can mainly get bacteria and archaea. I made some extension for viruses and even plasmids, but they are a bit more in development modes. But yeah, there are pipelines also for that, which kind of extend from the atlas, from the base atlas pipeline. If you want to look, you can look at the package, how to say, GitHub.com metagenome-atlas, and then look for other pipelines in the same repo of the GitHub repo. Then Maxim is asking why eggnog versus Proca. Eggnog mapper is really to annotate genes. Proca really makes the whole annotation of a whole genome, and usually it takes a lot of blasting, which takes I think, yeah, it takes a lot of blasting to several different databases, and this makes a bit less scalable, because often if you can get hundreds of max from a data set, and then time is still important. I think for gene annotation, I'm looking forward what Martin and Milot are to tell, because with MMSec it's really a tool which makes faster gene annotation, and yeah, looking forward. So the reason why I don't use Proca is mainly because the blast step and then installing of all the databases is complicated, but in theory it's simple to add in as addition. Yeah, if you get the genome sequence, you can use Proca to annotate them afterwards. And Maria is asking a question about the quality of marks of phyla from different abundance. Let's try to explain this by how to say. Let's go to the slide of the quality. So here will be the okay. Can I share? Yeah, share again. Okay, so Maria has asked a question about the quality of marks from different phyla which have different abundance. So what is used for the binning to generate the marks is based on abundance. So usually you take all the contigs of a high abundant species and group them together, more or less. Of course, the algorithm is a bit more complicated, but you try to group together contigs which have a similar abundance. And so of course, if you have a higher abundant species, then it's easier to assemble at the first point because you have more reads, and it's also easier to group them because you have more abundance to show. If it's very low abundant, it's very difficult to assemble. But at the end, if you have a genome independent of the abundance, at the end you can estimate the quality. So the quality is more or less independent of the abundance of a genome. Of course, yeah, from low abundant genomes you get generally lower quality than from higher abundant, and this is kind of the trade-off in metagenomics. If you need you need a lot more reads than, for example, 16S to get coverages of different genomes, and if you want to look at the low abundant genomes, you need even more coverage. So it's kind of a trade-off between what you get out of it and the price. I hope you are advancing with the tutorial. If you have any questions, then let me know, and I try to answer them.
Hey, Silas, there is also one question about the Python version. Do you have any comment on that? It's like people having problems with Python version 3.7 and 3.8. But the tutorial I tried running was with 3.6 and that worked. It's the question of Teresa in the chat.
So yes, for the Python version, ideally, I think with 3.6, it should work. If Atlas is not installed by default with 3.6, you can append it. Let's try connect for installing the, how to say it? Because I can try to answer it, but the idea is to append Python equal 3.6 at the end to install it. But I think I answered directly in the question.
Maybe you can show it in a terminal how to do it so people know what to type also.
So this would be the command to set up Atlas in a new environment. It definitely works on Mac and on Linux. I'm not sure on Windows per se, but that said, probably to install Atlas, really, it's the idea to install it on your cluster. You can do it for the tutorial. I think the test dataset is small enough to be run on a cluster, at least for the first steps. And yeah, at the end, you will need to go to the cluster. There are other questions. So yeah, for viruses. No, it's something I do. Yeah, for Virome, again, I think I have made an extension for the metagenome-atlas, and it uses Vibrant as main part. So this would be a pipeline to look for it. And then, can you use metagenome-atlas with human genome data? So it's really to analyze the metagenome or the microbiome of human. You can use it, and there's a way to provide the human genome to it. Yeah, so it's for the microbiome, and you can use for the human microbiome. Does it work on Mac? So I started using it in Mac. So you can do the tutorial, at least part of it. No, the tutorial you can do anyway on Mac. You can install metagenome-atlas on Mac, but I think there are some of the dependencies later on, which, for example, the quality control, we cannot be installed on Mac. So this is kind of some of the risks, or let's say the disadvantages. But yeah, anyway, you will need a lot of memory. It's like 250 gigabytes for the assembly if you want to really try it on a genome. And the tutorial is made in a way that you can look at the output, you can do this analysis without installing metagenome-atlas on your computer. So the idea is really to have a look at it. For me, it was often interesting to look what the pipeline is producing before then installing it. So that's why I made this tutorial in a way that we start looking at the output first.
Silas, can you look in the presenter chat, please?
Yeah. The questions in the other chat, you mean? Or,
I mean in our internal presenter chat.
Yeah, okay.
So from what I understand from everyone who is struggling to still install metagenome-atlas, in the tutorial environment, you don't need it at all. You only need all the plotting libraries, etc. If you're still struggling to install it, please stop with metagenome-atlas for now, but start the tutorial environment instead.
Yeah, the tutorial is made more. It's yeah.
But there's more of a downstream analysis what you can get out of metagenome-atlas from all the understanding and deeper insights that are revealed by it.
Hi, Silas. So we go over the tutorial, perhaps because I see some questions still popping up about the installation, but maybe it takes us too much time to look into this Python version 3.6 and 3.7 now. Maybe we can solve it via GitHub or so. But I also see one question from Laura asking whether finding genomes of viruses and fungi can work with the genome taxonomic database, which also include fungi. For me, I have tried till now only bacteria, but maybe still, as you have some comments to that or addition.
So I think for binning, I think it's really complicated to get the eukaryotes genomes out of it. I think the binning algorithm still, until you can cluster them together, group them together. But I think I haven't seen much publication really getting genomes from complete eukaryotes out of it, because they are just much larger. You have different chromosomes and so on. So for virus, there is an extension I said. For the eukaryotes, also the genome database. No, eukaryote genomes, if they would be binned together, then of course the quality control would kind of classify them as low quality genome, because it's a low quality bacteria. I think for eukaryotes, I will let speak Milot a bit more, because he has a lot of the tool to get genes from eukaryotes out from metagenomes. So I think I will let more time to explain it. Metagenome-atlas, no, it doesn't really work for eukaryotes. For viruses, there's an extension, but not so for eukaryotes. For the installation, yeah, I'm sorry. I think we really try to make it as simple as possible. We know also that, for example, Windows is not so good. But you can try to make a big part of the tutorial instead online. Yeah. So if you are here, just okay. Can I stop sharing the screen? Okay, here we go. So if you want to have a look at the tutorial, so as again in the first part is trying to look at the output, and at Atlas I generate now a cool report which shows all the output you can get from metagenome-atlas, the list of bacteria, the list of max you can get out of it with the species. You see here many of them can be annotated to species, but sometimes it's just this called CAG. Meaning that there's another MAC which is published some time ago, is similar to this, so they will classify it to the same genus but not the same species. So it's kind of a new species. The plot of the quality we have already seen. Well, the abundance of this. What's interesting is always to look at the mapping rate. Here we have still a mapping rate of 60%, which is quite low. Ideally, if you would add more samples, you will get more genomes out of it.
Silas, can you increase font size a bit? Zoom in a bit so it's easier to read.
Of course.
And maybe maximize the window, and then.
Okay, here will be the big screen. Okay, so
Great.
make it bigger. Thank
Great.
you very much for the
No,
comment. So we have the mapping rate. For the statistics, which is more important in the other part, there's an interesting paper about how to analyze microbiome data in general, because we actually, because to the sequencing, we can only get kind of the fraction. We can only analyze the fraction between samples. Usually we think we have counts, we have kind of the absolute abundance, but it's not really true. And I encourage you to read this paper. I don't think I have time now to explain the contents, but the kind of using this type compositional data analysis makes it much more easier to do statistics. And here, as again, to draw the PCA is also based on this attrition distance. So here we have the nice PCA. We see already two groups, the obese and the lean mice. Yeah, and then just a plot about relative abundance. Again, for the functional annotation, we map the genes to the genomes, and then we multiply the abundance of the genes by the abundance of the different genomes containing a specific functions, and with that we can get this kind of an estimation of the abundance of a function. But a function is always linked to a genome. You cannot really quantify functions as such in a metagenome. So this would be the output. We can already look at it. And then we are going back to the tutorial, which is here. Then you can ask for some questions. For example, we look at the example folder. I have downloaded it, or of course I have already. Here. So the folder looks here, the example folder. And we see here, for example, we have the genomes inside, or the FASTA sequences of the genomes. You can do whatever you want with them, pass it through Proca or other tools. So here we have 60 genomes, which are representative of a species. I said that there's this dereplication step, which groups genomes or bins together from the same species, and here we have one representative with the best quality per species. And yeah, you can look at the taxonomy, which we did before. Look at the tree. We have just wanted to say there's a gene catalog as well. So we have here the genome, the taxonomy of the genomes. In the gene catalog, here we have the raw genes. I think, yeah, I can open them. That shouldn't be too big. Yeah, gene 1, gene 2, gene 3, and so on. You can also, again, try to annotate them differently if you don't want to use EGNOG Mapper, and then make the link between the genes and the genomes. So, which is interesting. It's more for the second part in Atlas. If you start a new project, it generates a config file, which is just a normal text file, a YAML. And here you can define a lot of parameters. You can easily change how much memory are used for different steps. You can add the genome of the host, for example. You can change different parameters. But yeah, there are more explanation in the documentation. But the idea is you can really also customize a lot of the steps in Atlas. And then for the second part, if you want to have how to really do statistics of the mice, you can look at the notebook. You can do this even if you don't have everything set up. I think there's really the code you can run. It's more or less you can, if you manage to set it up, you can run it. You see that the mice are more obese. We then look, okay, there's a big change between these families. We have in the Joe diet, we have more Murispectacea, and the other we have Lachnospiracea. So we see already changes here between these two groups. It's more, yeah, to look at an output, but the more interesting thing is what you can get out of Atlas. So I don't think it makes much. So yeah, you can look at these two notebooks even if you don't have everything installed. You can just click on the link on the tutorial web page, and yeah, have a look at it. If you cannot install Atlas on your computer, it's not so important. The metagenome-atlas is really intended to work on a cluster, so ideally on a Linux. I don't think there are clusters of Macs. And the tutorial should work again on Macs, and even where you can install everything with Conda. So I'm sorry for the one which couldn't install everything. I still hope you can look at the output at the tutorial even without running. And if you try to install metagenome-atlas for your project, then it will work. Also, if you have questions, of course, I will be there for solve issues. Usually people write on the GitHub, and I try to answer them as quickly as I can. And during probably the week, if you have questions, you can use the chat on our landing page.
Silas, I see one question, because now people can run like Atlas run all, but is it possible to just run like parts of your pipeline, for example, only binning or not binning and so on? Do you have examples of code or parts that you add to your command?
Yes, you can run only parts of it. How to say it? So here we go. Obviously, you need the assembly step before the binning, and if you don't have quality controlled reads, you need the quality control read, the quality control step before assembly. But each of these steps can be run independently. So you can start, QC or Atlas run assembly. I think this I can recommend to you. If you have the test data sets, just run the assembly step. There also you can also run the gene catalog step independently. So if you have a metagenome is too complicated to really assemble and get max out of it, you can just run the gene catalog. In the documentation, I have really a step-by-step of these intermediate steps which you can run. So the idea is each of these steps you can run independently. And for the tutorial, probably it's best to run just run assembly.
Silas, you're still on screen share.
So yeah, maybe you want to start soonish, Milot. For people still have for people it's not cannot install it. The metagenome-atlas, I really encourage you to go to the either the documentation or which is the link at the end of the tutorial. And I really make step-by-step how to install the Mamba, which is a faster version of Conda to set up the channels. So then, so there's 2 steps needed before you can read the Conda install command. And yeah, otherwise it's a tutorial.
Silas, let's just make a 5-minute break or like a short break, and then we will start with our part.
take a short briefer, take a bit fresh air, and then let's start with MM6. See you all in like 5 minutes.
Okay, let's get started. Can you hear me? Does it work? I hope I unmuted. Perfect. So here's the second part of this tutorial, and this part is now about MM6.2, which is in comparison to Atlas. Atlas like a framework that is more on the genomic side. It's MM6 is really like focused on the gene side. And here you can see all of these like cute little characters. These are all part of the MM6 family. Let's hope. And so MM6 started in like 2017 with a search method for proteins. And this search method, to build that search method, we started to build like a certain amount of like modules and workflows to make this search really efficient, and also to make the code really reusable. And based on these models, we started like more and more to building up and more software. So for example, and next was like LinClust, it's like a linear time clustering algorithm, which we will see today how to use it and what to do with it. And then a protein level assembler that can assemble many folds more proteins from really diverse metagenomes, but also quite different tools. For example, metagenomic recently published, and is a tool that Silas was mentioning. If you're interested in eukaryotic metagenomics, metagenomic can help you to get proteins out of these metagenomes by finding the exons and building up these protein models and extract them. Spacefarer, not yet published, only available as a preprint, is a tool to detect phages, more sensitive and more reliable. And we have an MMSeq server that you can use online without installing MMSeq to just search search proteins against multiple databases. We will also present it later today. And so for this, the required software for this one you can install with Conda. We already pre-installed it here. You only need to install MMSeq and Plus. I hope that works for most of you. You probably have issues if you have a Windows computer.
Yeah, it doesn't work on Windows. So you can follow, you can just watch us. We will execute all commands and explain what's going on. If you want, you can download this Jupyter notebook and just execute the same commands and run along, but you don't have to. So just whatever works for you. Wait, let me mute myself again before I give you control back.
Yeah, so this command here now sets the path variable in the Jupyter notebook and executes some bash script here. So this exclamation mark means that it executes some bash script. This is like source activate tutorial, which gives you back a path. And then we just set that path to the environment so that all of the tools are now in the path and can be used by our notebook. And let's start with what we're gonna do today. We have that case. So first of all, this is work that was not done by us. We will later disclose who actually did this work. For now, we'll keep it secret to not spoil the adventure. We start with a patient, and this patient is a 61-year-old man that has headache, claims to have headache and confusion, and suffers from this for some time. And he goes to the hospital, and in the hospital, they figured out that there are two like tick bites on his body that are 2 weeks old. He is a woodworker, so his occupation leads to a lot of interactions with rodents and bats and ticks and all of these things that can transfer pathogens. So after some time, his situation is developing really badly, and he gets weaker and has more problems, and headache gets stronger. So they did MRI, and in the MRI, they found some inflammatory region in his brain. I don't know if you can see my mouse, but it's this white area here. So if some inflammation that occurs. So it's some encephalitis, and they don't know what is really causing it. So here for this tutorial, we try to figure out what is actually causing this disease. And there's this here is a picture from Trish Simmers from a publication from 2018, and she compares pathogen detection methods. And on the left side, you can see the traditional workflows. So you have this different types of specimens you extract, and then based on your pathogen that you assume that causing this disease, you do certain kind of tests. And these tests can vary from like days to weeks. So, and also you need to know what kind of test you want to do. So doing all of the tests is really expensive and not really doable. On the right side, you can see a workflow that is updated and done now in a new fashion, where you would do the whole thing with like next generation sequencing. And in that paper, they claimed it takes up to 3 days turnaround time from sequencing to diagnosis. But this is all based on Illumina sequencing. I think with like newer technologies like long read sequencers and nanopore, you can get that time significantly down and probably under 1 day and figure out what's causing it. The advantage of these NGS workflows is that you don't need to know what kind of pathogen you're searching for, because you're searching for all of them at the same time. So because you just look at the DNA of them. And these workflows, and so here's a workflow how this works. So you have your patient or the animal, you want to figure out what kind of pathogen they have. You sample from there, you take some specimens from the, for example, spinal fluid or some autopsy from the site that has the inflammation. You then extract DNA or RNA or both of them. And at the same time, you also do this extraction on some kind of control. So you take some just normal water and do the same extraction of this DNA. Then you put all of these samples in a sequencer and you end up with short reads. Nowadays, still most people use the Illumina sequencing platform, even though the nanopore might be faster, but it's just the throughput is much higher still on Illumina, so it's cheaper and the error rate is lower. So that's actually the reason why we actually do that. And then if you have that, we start searching these short reads against reference genomes or reference proteins to produce some kind of taxonomical classification of that sample so that you can see what is actually in your sample. And here what they did is they performed some biopsy on the brain on day 8 and sequenced that and ended up with 14 million short nucleotide sequences. And they removed then from this short sequences all human. They use Kraken for that. In a minute, I will explain what that is. And now the question is, why did they, so they uploaded this set and we're gonna work with this set. So the question is, why did they remove the human from this reads? And yeah, if you work in this clinical setups, you have to be very concerned about privacy. So you don't want to reveal the genetic identity of the patient. And therefore you remove all of these of these reads first. So Kraken works in a way that you start with a read. So here this query sequence would be the read. And you break that read in overlapping short words of length 31. So here's each of these boxes is a k-mer of length 31. And then you match these boxes to a database. And this database is a pre-computed k-mer table of our reference genomes that we have. And we can then, for example, find here these orange boxes are E. coli, or this red box is Shigella. And like based on all of these k-mer classifications, so you have like nearly the length of the reads minus k and the length of the k-mer classification. And based on this, you do some voting. And based on this voting, you decide what kind of taxonomical label you give to that read. And this is really fast. So these methods work really, really fast. And they're also very specific, but they have some drawbacks. And now the question is, what do you think are the limitations of nucleotide-based classifications? So one problem with nucleotides is that nucleotides evolve a lot and fast. So sequences evolve quite a lot, and you have maybe a resolution to 80% sequence entity. You can align things up to 70 to 80%. This is actually the lower end of what you can still detect on the nucleotide level. And you can reach this threshold actually fairly fast. And it can be that for pathogens, we do not have any close relative genome or like related genome in the reference database, and therefore we cannot really match our reads back to this reference. So there's a way how you can overcome this problem of close relatedness. And this is, instead of like using nucleotides, you can use proteins. And proteins have that property that they are conserved over really a long period of time. So for example, you can see here two structures of ubiquitin. One is from human, one is from yeast, so really far away. And the sequence still is a bit similar. So actually you can see similarities, so colors meaning similar amino acid properties. And you are able to align these sequences even over these billions of years, and you can align them up to like 25% sequentiality, so really, really far away. So how do you do that now? how can you take nucleotide reads and find them in a protein database? And the answer is 6-frame translation. So you take your read, here's your read, and you look at all possible reading frames. For example, here are the 3 possible forward frames, and then you do the same on the reverse frame. And you translate them into amino acids, and you match these amino acids back to the proteins. And only the frame that actually encodes a protein will be the one that matches something. And often the frame that has no stop codon in a certain range is the one that is really actually the coding one. For example, here, I think the 3rd frame would be the coding one because there's no star. So most likely probably the coding frame. So what makes MM6 like well suited for doing this kind of searches is it has a very fast pre-filtering stage. And that's really important because often you search with a very big query set. So you have millions and millions of these short reads, and you want to match them against really big reference databases with millions of sequences. So as a first step, you have to like prune down the potential candidates of hits very fast, because at the final step, MM6 does some kind of alignment, so Swiss Waterman alignment, and this alignment is not that fast. So you cannot align your read against all of these sequences. So the prefilter has to reduce the space. So MM6 does this by searching k-mers on a diagonal, double k-mers on a diagonal. Once they find it, they compute an ungapped alignment. And if this ungapped alignment score is above a certain threshold, we do an gapped alignment using Swiss Waterman. And this performs really good. Here on the x-axis, you can see a sensitivity. BLAST is on the sequence search methods the most sensitive, but BLAST is among the slowest. Swipe is a bit slower. Swipe is actually a fast Swiss Waterman implementation. And here on the y-axis, you have the speed up relative to BLAST, and you can see that MM6 here, this red one, can be fairly sensitive and still like 70 times faster than BLAST, or really, really fast, 100,000 of times faster than BLAST, but not really sensitive anymore. So with this, you can actually set up your sensitivity and your speed trade-off based on your use cases. So what do you do when you cannot find, if you find an alignment like this? So you have these 2 sequences aligned, and the e-value of it is 0.2. So the question is now, do you think these sequences are homologous? And normally, if people ask these kind of questions, the answer is always yes. And yes, they are homologous. And what you can do is you can take that sequence and you can align it against a multiple sequence alignment. And by doing so, you will see that the sequence here has these conservational patterns. For example, this green here is really well conserved also in the sequence and in the MSA. And based on this conservational pattern, you can actually re-estimate the significance, and you can see that they are actually homologous to each other. And MM6 is also able to do this kind of profile searches, and actually only really fast tool that can do it at a metagenomic scale, and can compete with methods like PsyBLAST. So with this, I will hand you over to Milot, who will now guide you through how you can use MM6 and how like the first part of the tutorial. Okay. I will be in the chat.
Yes. Hello, everyone. Wait, yours. Okay. No, you're still controlling my mouse. Okay. Oops, that I didn't want to do. Let's share again. Okay, let's walk through this thing. When you install MM6 of Conda, it's a command line tool mostly, and all commands from MM6 share this common pattern, where we have as the very first word the MM6 binary, and then one of many commands, and then you pass it multiple files or multiple databases, and optionally some parameters in the end. Over the years, we have built many workflows and many modules that do these searches, from nucleotide searches, nucleotide protein searches, and all kinds of other combinations, as Martin mentioned, the profile searches and so on. We do taxonomy assignment, whatever. All of these we've wrapped as workflows that give you basically plain text faster input, or compressed faster, and return also instantly readable downstream processable FASTA file again. or some clustering, etc. We have modules that give you more flexibility, that stay in this MM6 pattern where we produce a database of queries. In database, we mean something that where you have a large set of queries, like in a metagenome, millions of reads, and each of them are one entry in the database, and then MM6 is able to massively parallelly process these large number of reads or contexts or whatever you have. If in our main, in our main workflows, you manipulate those in some way, create them, manipulate them in some way, and we have built over time a wealth of smaller modules. You can see, I think currently it's about 100 or so that do all kinds of different things with them. And all the methods we publish is basically a new combination of any of these modules with just a few new ones added for the next tool now. And with, in the tutorial now, I want to show you a bit more how to use these tools to get the most out of your data and how to be efficient in exploring a new set. Okay, let's get back to our patient. We prepared these reads for you. They were, we downloaded them straight from SRA, the sequence read archive, and ran them with Flash, read merging tool, a pairwise read merging tool to merge those that overlap into one read to increase our signal slightly. So let's download these reads. This is a not very large file, I think like 30 megabytes or something. And it contains 200,000 reads, about 200,000 reads. Another thing that MM6 needs, then, as Martin showed earlier in the slide, sorry, that we always search against something. So we need a reference database. In this case, so it will run on also on your notebook, for example. This set of slides is running on my poor MacBook from 2013, so it's not the fastest horse in the stable. We are using a SwissProt. It contains 500,000 proteins that are extremely well curated since they're manually curated. Usually, what you would do as a user, you would use our MM6 database command that would download one of many databases, such as here in the SwissProt. But we are not going to do that today, since we want to have reproducible results for every user here should later identify the same virus and so on. I'll show you in a second how to build this from a database we already prepared for you. Okay, let's do that. I've ran these few steps already earlier before, since they take about 3-4 minutes to finish, and I don't want to spend those 3-4 minutes at this point. If you have a slightly faster notebook or you're running it on your workstation, all these steps will be pretty much instant. And what we see here, we download a FASTA from a certain year. It's an older release. We download a tab-separated value file that contains a mapping between the accessions of this FASTA file to taxonomic identifiers in the NCBI taxonomy. And then we create a new MM6 database out of it, with this create database format. So remember what I told you earlier. Here, what happens is for each entry in the FASTA, we create one database entry, and then MM6 is able to access this parallel and therefore very fast and use the most resources. Use all the resources you have on your system or even on your cluster. And then we add this annotation so MM6 can read it and prepare this database in such a way that searching against it multiple times remains fast. This last step is optional. Okay, so let's continue with MM6. what we see now is all kinds of parameters this one module, this one command will take. And then we see that in 4 seconds it processed those quite a few reads and converted them to the internal format. You might wonder why are there 400,000 reads here and not the 200,000 I mentioned earlier. Maybe take a few seconds to think about why that might be. Okay, the answer is that most of these reads were not actually these are paired end reads, and most of them were not overlapping enough to be mergeable into one single read. So we just left them unmerged to be able to still process this, to do something useful with them. Okay, one of the more recent additions of MM6 is now to be able to assign taxonomic labels to reads. Let's run this. We add one optional parameter here. This is the sensitivity parameter, as Martin showed you. Let's find the slide. Oops, that was a bit too far. As Martin showed you here, as this taxonomic analysis is now running on my old notebook, we don't actually want it to use so much resources that it will take too long. We are fine enough with low sensitivity searches. This somewhere in between here. Okay, let's see how far we've progressed. This tool, oops, let's close this. This tool is now progressing, and MM6 is calling one of all of these internal modules that I showed you, one after the other as a workflow. So here we are calling the search. Oops. I see a black screen shared. Martin, do you see me?
Yeah, I see you. So I just asked the operator about this black screen issue. Somebody seems to have black screen issues. I hope that not everybody has it.
I can restart sharing. Maybe it helps. Stefan, did this help? Do you see my? Do you see the? Do you see the screen again? Okay, great. Okay, so what we see here now is MM6 calls all these internal modules. The one that Martin mentioned earlier. With nucleotide reads, we cannot compare them directly against a protein database. So we have to extract putative protein fragments out of them through the 6 reading frames. We have one module that does this part. And then we have this fast pre-filtering stage that Martin mentioned. After this putative fragment extraction, we create about 1 point something million fragments, and then search this 1.2 million fragments against 500,000 entries in the reference database. This takes about a minute or two. It would be slightly faster on a faster device, but MM6 is good enough to scale down from basically a phone up to a huge compute cluster. I've actually ran MM6 on an Android phone before. It works. The next step is this slower alignment stage. Here, what we see now, instead of running now this huge search of 1.2 million sequences in a square against 500,000, the pre-filter has rejected most hits. So for every one of these queries, on average, we only have to do about 200 alignments. This is now not a huge problem anymore. If you were to do this with BLAST or something, you would take forever for a search of this scale. But here we run through all these alignments in about 41 seconds instead on an old notebook. This stage is not so important. Then after we get all these alignments, we can do some taxonomic assignment based on this mapping we extracted earlier. Oh, sorry. Okay, we're through. Oops, but I didn't want to do. Let's go. Now what we see, we have, as we requested here in the command before, we created one result database. But this result database is not super useful to look at as a human because it contains some binary data and so on. We have a module that converts the result into a human readable format. Let's look at this. Here now we see the first 1,000 lines of this file. For every read, we now have an assignment or an unclassified assignment. Most of them remain unclassified. A few of the reads have some taxonomic hit. So what we see here is the read accession, the taxonomic identifier of that we assigned, the rank of this identifier, and the name. So the species, somewhere further down you will see a species. So why do we see that most reads here remain unclassified? Well, two things. First, we search with low sensitivity, meaning to save my notebook from overheating, we search with low sensitivity. So for some of the hits, we just won't be able to recover any hit. And the other thing is, this is genomic data, so we will find a lot of intergenic regions and well, whatever else remains. And these you will not expect any hits anyway. So it's okay with this, but a lot of this remains unclassified. Let's do some preliminary analysis before we move on to having some more in-depth visualizations. Here I made a simple bash pipeline that extracts now this column, the last one, the names, and gives me the most the ones with the highest count. We see that most of the reads remained unclassified. Then we see a few weird bacteria. Some, when you look them up, are either in soil or attack tomatoes, which are already odd. Some virus. You also don't expect to find much viruses in a brain biopsy. E. coli, you see some human reads, and so on. Oops. So why are they? One surprising thing is that there are still 700 human reads here. Might this still be a privacy concern? Well, maybe. One reason is that Kraken might have just not been sensitive enough to find them. The read quality, as we will see later, is also not very great. So they might have had some sequencing errors or whatever. And Kraken was then not able to recover them anymore, or well, there might be many reasons, but remember that safeguarding the privacy of a patient is not an easy task. Okay, what's with this weird bacteria? Do we expect a tomato plant attacking bacteria really in the brain? It's difficult to say, as Martin mentioned earlier. In a lab setting, in a pathogenic lab setting, we would have also a control set. Authors sadly did not give us that. We only have the set of the sick patient, and so we cannot really do a differential analysis to compare. Is this really there? Should this really be there? For now, we can only suspect that this might be a different contaminant or so on. Okay, MM6 offers you many different ways to look at the same data. One thing is to get a report that is compatible to Kraken. Here you will see a hierarchical view. You will see a hierarchical view of the data in a second. Okay, we see the still same numbers as before. Oops. Let's look at this thing. So what we see here now is basically a textual tree view that starts from a root and then is intended every time it's on a slightly deeper rank in the taxonomic tree. You see the relative the abundance among the whole set percentage. You see the count of the reads that are assigned here. Don't quite remember what this was. I think it is a unique number of reads that are assigned at this rank. The rank name, the rank identifier here, and the actual taxon label name, and so on. And this goes down and down and down. This is still not very useful to look at, but we can use one of the mainly useful tools that work with Kraken output, such as Pavian. Oops, I want to go tutorial. Pavian is a really neat tool that gives you nearly publication-ready graphics for visualizing the content of this taxonomic profile. We see this similar view as we recovered with our bash commands now. Again, we see this some virus, something that we really don't expect in the brain, but also other things, the same other things that you don't expect in the brain. Okay, and there's a third view that I want to show you how to.
Can you go back? I just
Yes.
want to explain a bit more.
Oh yeah.
So Pavian is actually really great, and it will make your analysis really, really nice. So you can, for example, filter by taxa here in this upper field that Milot highlighted. And we filter, for example, already out the chordata and artificial sequence and others. I mean, chordata, we don't really expect to have something with a spinal cord in our brain, so we don't want to look at them. So you can really remove things that you don't want to look at. And this is really just a sample view. If you have multiple samples that you have a control and you have your positive set, then you can compare these in a table-wise manner where they already do some like set score statistics to find if the abundance between these taxas is big enough or small enough, so you can pre-sort them and actually find maybe very comfortably already if something is a pathogen in that way. Unfortunately, the comparison view only works if we have multiple samples, so we cannot really present it at that point.
Maybe we want to take a look at the sample they have. It's unrelated to the virus, but just as a short example what you can do with a powerful tool like this. Also in combination, you could as a downstream tool from MSX or Kraken, for example. Here they have data from some brain biopsies. Oops, does this work?
Yes, it should work. You have to wait a bit. It has to do a computation of the statistics.
Should I look at result overview instead? Did I do it correctly?
I think you have to click on the example HMP. Okay. Yes, and now you go to the comparison view. And
okay, I don't think it likes it. Anyway, let's move on, since we maybe it's just a limitation of this how I embedded Pavian into this Jupyter notebook. Anyway, it's a very useful tool for your work with metagenomic data. I have a third option to look at composition, taxonomic composition of metagenomics, and this is in form of a Krona visualization. This is a circular view, and MM6 can produce with the same, pretty much the same command as before, just adding this other report mode. This report mode produces an HTML file. It can produce this very neat and useful visualization. Let's look at it. So now we have a third, the same view on the same data, but in a different way. We see again that the vast majority remains unclassified. Then we can switch here and see these abundances. Okay, when you if you spend some time now looking up most of the common bacteria on Wikipedia or the NCBI of the supposed pathogens in Wikipedia or in the NCBI taxonomy or so on, you will find that most of these things are very unlikely to cause encephalitis. So the only option really that we have here is this tick-borne Povazan virus. And for this, oops, let's open this. For this tick-borne Povazan virus, we see that we find about 1,200 reads, or we will see that in a second. Looking through this manuscript, they found, Martin, do you remember what was the number? How many reads did they find again?
230 were classified with Povazan using Kraken, so searching against the nucleotide database. Yeah.
So apparently this virus was not that easy to find, and they did the whole diagnosis in 3 days based on 130 reads out of 14 million, which is also pretty amazing. When they actually got the lab result a few weeks later, I think like 3 weeks later, it was confirmed that this is indeed this was indeed the pathogen, and they could at least try to start some treatment very early. So after day 11 of the hospital visit, they could try to start some treatment. Okay, let's go back to MM6. The same thing here that I mentioned very shortly earlier before is that we can use one of these easy workflows where we get some faster input and a reference database, and then only plain files as output. I'm not going to run this now because it will take again like 3 minutes or so, and I don't want to spend this time. But I have this, I already computed this before. This is one very useful thing that MM6 will return for you. Here we have a report where we look at our data in a transposed way. Here for each target in the reference database that was hit, for each target in the reference database, this protein, for example, we see how many of our query reads hit it. And we see some measures like, oops, I want to see this again. We see some measures like at the coverage. We see that this one protein was hit by 1,244 1,244 reads. And we see, again, the same information as before, the taxonomic identifier, the rank, the name of the species, and the whole lineage. And we see some coverage information of how much of this protein were covered by this 1,244 reads. So we have, so on average, this one protein was covered about 12 times with all the reads. Martin, you're an expert. You're an expert in pathogens. What do you think? Is this good or bad? It sounds kind of weird, right?
Yes. So what you would expect to see when you have a virus, and you really want to verify if it's really a real virus or not just a technical fragment, is you want to see multiple protein hits. But here there's only one hit. I mean, this one protein is very well covered. It's like 73% covered. So nearly the whole protein is covered, but also the sequence entry is nice and has a 12x coverage, but why is it just one? I mean, there should be multiple ones.
So now we get some interesting avenue to investigate further. And if we look at it, we see in NCBI that this virus expresses its whole genome as a single protein, as a polyprotein that is later cleaved in its functional subunits. So actually, even though initially you would expect this to be a bit odd, you see, okay, this looks like good evidence. Let's continue. Let's try to form some plan of action for the patient's care based on our analysis. Okay, so last task, I want to show you a bit of how these MM6 modules plug in together by getting now this taxonomy result we produced, and producing again one FASTA that contains only the Povasan reads. So we need this taxonomic identifier, this 39008. Well, here. And we will use a different MM6 module that can subset this LCA result based on a taxonomic identifier. Let's run this guy. This will take a few seconds. This is a step that we can still improve in the future, hopefully. And produce then in a bit of a roundabout fashion with our MM6 modules the input we need to create a subset of this database of reads that we produced in the very beginning. So here we produce one file that contains all of the internal identifiers, database identifiers, and we will subset this database to here, and produce then a FASTA file. Now we have a FASTA file that only contains this pathogen. Let's see. We found all our reads. Okay. So what can we do now? Martin? What can we do now with 1,233 reads from one single species?
So, yeah, let me take over. Can you give me the control, please?
Yes. Let me use this.
Okay. Let me click on that. Okay. There are like multiple ways how you can go from here. Like normally you have to verify now that these reads are really correct. We did several steps. Milot showed that there's this protein that contains all of these like multiples, all of these protein and like this protein that contains multiple proteins, encodes multiple proteins, and that this is really pretty much covered. So as a next step, what we want to do is actually want to see if we can recover the genome. To see if there's maybe something special about the genome, and also maybe then to get more evidence and to actually look at what's going on with this pathogen. And so assembly, as Silas explained before, on the nucleotide level is you take your nucleotide reads and you break them into k-mers and connect these k-mers to a graph, and then you can get a very messy graph sometimes, or sometimes you get a very clean graph, and then you find a way through that path through that graph, and this represents your genome. And these de Bruijn graph assembler are de facto the standard in the metagenomic analysis, like Metaspades and Megahit are really widely used and work really, really good. But they have some disadvantages. So in the past, assemblers were actually overlap assemblers. So the cellular assembler that was used for the human genome tried to find overlaps between reads. And then based on these overlaps, they built some kind of overlap graph and then reconstruct the genome based on the overlap graph. But there were like many computational issues coming from there with to keep this graph in memory, and also finding the overlaps is more expensive than breaking reads into k-mers and building a de Bruijn graph assembler. So in this section, we're going to talk about PLUS, which is an overlap-based assembler, which not just assemble it on the nucleotide level, but instead assemble it on the protein level. So it tries to find overlaps for proteins. Somehow I cannot click. Now it works. And so the question is, why would it make sense to assemble on the protein level when you actually want to have the genome at the end? And one reason why actually a protein assembly still makes sense is that in these metagenomes, you have this huge diversity, especially in diversity in sets like soil. The diversity is so high because there are so many different strains of this bacteria or of this virus, and they can mutate quite a lot in these wobbly positions without changing actually the underlying amino acids. And this makes it really difficult for this de Bruijn graph assembler to assemble. But on the protein level, mostly a lot of these SNPs collapse and you can find overlaps. So there's this advantage coming from there. And with PLUS, we compared ourselves with PLUS. PLUS actually really just assembles proteins, nothing more than proteins, no genome that you get there. But we compared ourselves with the state-of-the-art metagenomic assemblers, megahit and metaspades. And how did we do that? We took these metagenomic reads and gave them to megahit and metaspades and let them assemble it. And then based on this context, we called genes. And we compared these genes to PLUS. And here on these plots, you can see on the y-axis the assembled amino acids. So how many amino acids. On the x-axis, you can see sequence entry thresholds. And these are the thresholds that we use to cluster these protein sequences. And we can see across multiple environments like human gut, eukaryotic ocean transcript, or soil, we get more protein sequences out of the samples. And this has often to do that pretty much the diversity is too high and these other assemblers cannot even produce a contig that encodes them. But today, we will use some software that is not yet published that Annika Seidel is working on. We're pretty excited about that one. It uses this protein guided, it uses protein overlaps to actually reconstruct contigs and get genomes out. And the idea works similar. In the beginning, you use some kind of PLUS-based assembly. So you take your nucleotide reads, you do some 6' translation, you find overlaps between these protein fragments, and then you co-emit the nucleotide sequence. And once you have assembled these proteins from start to stop, then you have your assembled ORFs. And now the task is that once you have your assembled ORFs, you can try to stitch them together with these linking reads. Often in microbial communities, or especially for viruses, the genome sizes are very small and they're very packed. And often, sometimes even ORFs can overlap. So doing this kind of assembly, you can actually often just overlap these assembled ORFs, or if not, you just need very short linking reads that actually combines them to longer contexts. And we looked with this assembler that we unfortunately called PLUS Hybrid. At the time is still a work in progress title. It's not a hybrid assembly with long and short reads. It's a hybrid assembly between protein and nucleotides. So don't get too familiar with that name. It's not going to stay. We tested it on some sRNA virus metagenomes. So there's the sludge set that was published in this year in Science, where they took from different sites around the globe samples, and they tried to extract single-stranded RNA phages. And these single RNA phages that they found, they encode these 3 core proteins, these MP, CP, and RDRP. And so what we did is we also assembled all of these sets using our new assembler, and we compared the assemblers that we can get with our new assembler against megahit, metaspades, and RNA spades. And we checked how much of our assembly overlaps with these other assemblers. And actually we cover a huge fraction of the other assemblers, while they do not cover as much from us. So we get more genomes out of these environments using these kind of hybrid approaches. And we also looked at our contigs and how are the distribution of the proteins that we find on these contigs. And here you can see the fraction that was published in that paper, how these 3 proteins occur, and our fraction that we get out is very similar as well, which all look very encouraging. But we did multiple other tests, but this again is still work in progress, so just be careful. But we're going to use that now for assembling our Powassan virus. So PLUS again follows a similar structure than the MM6 module. You first have the name of the program, PLUS, and then you have the name of the module or like a workflow you want to call. For example, here this hybrid assembly. Now the next parameter are the PowerSun reads that Milot extracted before, this 1,200 something reads. Here is the output, the name of the assembly that you want to produce, and you need a temp folder where Plus can write its temporary files. You can set some thresholds. Here we set a threshold for min sequence identity for the nucleotides of 95%, meaning that the overlaps between two reads, so we compute these overlaps and the alignments between them, and they can not be below 95% sequence identity. Silas mentioned that before. 95 is this like species criteria, this universal threshold that can describe every species separation, which is probably not true. Yeah, by now it's still an definition that we use and work with. And the next parameter is the min context length of 200. So we don't want to see any contexts that are below 200. And this min length, still unfortunately named, is the minimum ORF length that we want to consider, and this is the ORF of length 20. Let's call that program.
Usually you wouldn't have to specify this parameter, but this set is a bit odd in a way. These reads that reproduce are a bit odd in a way that they're rather short. They're on average about 100 base pairs long. And Plus does not do very well at automatically detecting that, so we have to reduce our default size that is optimized for better reads that Illumina will produce. So usually Illumina will produce like 150 to 250 long reads, and here we have to manually specify this. Maybe hopefully we will improve this in the future slightly. Let me mute myself.
Yeah, scrolling is a bit complicated with the remote one, but from the structure, again, you can see that Plus uses these similar syntax and similar output to MM6 because actually built upon MM6 modules. Here, for example, it's the k-mirror matcher. Before we saw some createDB. So you can scroll to that and maybe you will recognize already some of the modules that you have seen before in the tutorial. Okay, so now we can have a look at this assembly that we produced. And so we will see that they are just not really long fragments. That's the first thing you already recognize. They are among like 300, 200. I think the longest we can find is around 500 to 600 long. So it doesn't seem like we can really assemble this virus properly, so we cannot really recover a lot of this virus. Oops. Okay. So how many sequences were assembled? So we can check that with grep. And we should get 46 sequences that are in this, that are longer than 200 that we could assemble. And in default, this hybrid assembler still produces redundancy because it takes reads and makes them actually longer. And sometimes these contexts look very similar. So this is not this 46 is not a representative number how much actually really was assembled. To do that, we're gonna look at clustering and dereplication. So we want to dereplicate that set so to actually know what we really assembled from this one. And sequence clustering works in a way that you have a sequence set and you want to find these green sequences here. These are the sequences that that represents a lot of other sequences in the set. So, for example, if you want to reduce redundancy, this would be like a sequence that covers as much other sequence in the set that fulfill your clustering criteria of sequence identity, coverage, and so on. Initially, we were building that software for proteins, but here I'm gonna use it for nucleotides, which is also more advanced recent feature that we got into and really work heavily on to get this nucleotide clustering as good as possible and as fast as possible. And the idea of this clustering in MM6 is based on LinClust, and LinClust is a linear time clustering algorithm. On this plot here, what you can see on the x-axis is sequences we clustered in millions. So it's from 0 to 120 million sequences. We took certain sets and clustered them. And we gave them to tools like CD-HIT and uClust, and these are proteins, and we clustered them at 90, 70, and 50% sequence identity. You can see that most of these tools actually have some kind of exponential runtime, while LinClust really pretty much scales linearly with the set sizes. And also we combined LinClust and MM62, which is, we do that for sensitivity because LinClust is really fast, but not as sensitive. But if you combine it then with an MM6 following step, which do an exhaustive all against all search, and then cluster the remaining sequences, you get very sensitive. You can be still much faster than these other clustering methods. And really the main advantage here is that it's linear in that remark. And so how did we achieve this linearity is, here we have these black dots. These black dots are sequences. Now we take 20 k-mers, for example, from these sequences. We don't pick them randomly. We hash all of these k-mers per sequence, and we pick the one with the lowest hash. Then we sort all of these k-mers. So we put them all together for all of the sequences. We put these k-mers in a long array. Then we sort this long array, and then end up with these k-mer groups. For example, this red circle here describes a group of k-mers, and all of these sequences within that circle share that k-mer, so have that k-mer. And now we select a center sequence per group. For each k-mer group, we pick a center sequence. And this property of that center sequence is currently only that it's the longest one. So this is really a really rough approximation for a cluster centroid, and it could be much better, but this is really fast. And it helps us to reduce the runtime, which we will see in a second. So now we align all of the sequences against the center sequence. And we check if the criteria are fulfilled. So alignment thresholds and everything is fulfilled. So sequence identity, coverage, and so on, e-value. And if this is the case, then we assign them, Then we assign them to this centroid. And this reduction to the longest one is actually the trick that reduces the runtime to linearity, because if we wouldn't do that, then you had to do this comparison where you do all against all, and then you figure out which one would be a good centroid, and then you pick that one. So then you end up with your final clusters. Okay, and EC cluster again is within MM6. Here, similar fashion, use an easy workflow, calling this easy cluster module. You give it the assembly from our new assembler, give it a name of the output, so assembly clustered, for example, then the temp directory again. You define some coverage mode and some coverage criteria. Let's execute that. And then, oops, yeah. So again, yeah, it calls all of these different modules. Here is this LinClust module is called. So this is a combined workflow that first calls LinClust, then afterwards an exhaustive MM6 search, and then it clusters all of these results. And once that is done, let's just shortly talk about coverage, because a really important topic that I often see in publications is not can lead to problems, or like I feel like have certain issues or implications. So MM6 has this coverage mode 0, 1, 2, and 3. 0 is a bidirectional coverage. That means that if you have a protein, you want it to be covered. You have both the member and the centroid to be covered at the percentage threshold that you defined. That is really great if you want to keep, for example, the domain structure of a protein intact. So because then the alignment is nearly stretching all of that, all the range of that protein. Mode 1 is a mode where you want to more or less the shorter sequence to be covered at a certain percentage. So here the target sequence has to be covered at 80%. The query it's not important for the query, only at the cover. This one is covered. So why would we use that for this assembly reduction? Is because if we assemble something and then we have like a short fragment that is somewhere within the stretch that we have assembled already, then we just want that to be collapsed. And so this is in general this property. And we have the same thing for the query as well, and then some mode with query length, which is not very often used. I think most important is actually 0 and 1. I think it's always good to think about which coverage mode do I need when I run clustering. Yeah, let's, you want to take over, Milot, and look at the results of this clustering?
Yes. So what we see now is we have now multiple files as output, namely these three. This is a file that contains like a pseudo FASTA, a FASTA of FASTAs basically with all the clusters. So cluster member and then cluster representative and then cluster members. Then we have an adjacency list of cluster accessions which also define the clusters, and we have a FASTA file that only contain representatives. Let's look at these last two. We have now one file that only contains representatives. We see that this accession 0 was a singleton cluster. 1 was also a singleton cluster, and 3 as a representative. It had one member collapsed. 27 was collapsed into this cluster 3, and so on. And here we see a representative of this cluster. These files are a bit longer, but we are only looking here at the first few lines. Remember, we had like 44 sequences, and we dereplicated them down to 17. And now let's see what useful things we can find out from our uniquely covering cluster representatives. We will now download this polyprotein from UniProt, just as a simple wget, and give it straight to MM6, which reads it from standard in, and we will search now against our 17 assemblies. So it's also a very small scale search, but it's still useful to visualize our results and find some insights from it. So we search one sequence against 17, and with a special output mode, we again get some HTML output that gives us the opportunity to interactively investigate this one sequence. Okay, let's see how, whoops, I have to run this first. This should take.
Just a remark. Normally, in default, when you run the easy search, it produces a tab-separated output that is similar to this BLAST tab format. But with this format mode, you can freely adjust the columns. You can add things, and now actually you can even grade this HTML output that Milo will present now.
Okay, so let's see how this looks like. And the very first thing we see, it's not that amazing. We can cover a large part of a protein, and we will see in a second what's up with these regions where we don't find anything. But still, we see for our best hits, we see that they match with very good e-values. Here, 10 to the power of -65. We see the alignment, which looks nearly identical, and so on. So we are pretty certain we found the right. We assembled and found good context. However, we still want kind of to know what went wrong. Why didn't we have one single contig spanning the whole polyprotein? For this, Martin has prepared this view from the IGV, where you mapped all reads back to the reference genome, and all these colored dots. We see that there is some variation, and we see also other weird artifacts, such as it's cut. All the reads we find are cut very cleanly to some regions. This also seems like it's not just metagenomics. There was some downstream processing biochemically happening here. And from all the variation we see, okay, it makes kind of sense that assemblers struggle to produce a good assembly. Here we see that MegaHit also did a reasonably good job to produce the same context. It is missing a few, which Plus was able to assemble.
Just a remark for this. Later, we will talk about what paper that is. But in that paper, they use hybridization as well as metagenomics to enrich that virus. So maybe what you can see here are hybridization fragments. So if hybridization is you have a certain capture probe and then you capture that virus. So for example, if you capture it always at a similar position, then maybe you get these hard cuts that are very artificial. Because if you would look at these kind of read layouts normally in a normal fashion, you would think that coverage is more or less evenly distributed. Sometimes it's biased because of GC or so, but it wouldn't be as extreme as what you can see here. So that is really something that is that artificially amplifies that region, but doesn't really look at these regions over here.
Okay. And as a last thing, after showing you all these things you can do with MM6, I want to show you one more thing. We also have a web server. Maybe I open it in separately in a view, where you can put in whatever protein sequences, nucleotide sequences, and search against a few big databases. We have a special database that hopefully contain that is very enriched for eukaryotic sequences. We have domain databases, structured databases, and the clustered version of a UniProt. And you can search against these very fast. I've prepared here one result for you already, and now for each contig that we found, what we assembled, we can now look at what structural or what protein families we can match. So here for this first contig, we can take a look, for example, at PFAM, and we find flavivirus glycoprotein domain. Oops. We find here with somewhat low sequence identity, we find dengue virus structure and so on. And we find also many references in the large reference database. Now you might wonder why we don't find this polyprotein since it is in the SwissProt. This uniclass 30. This is a cluster database. So what I assume is that the polyprotein was clustered into some other representative, and now we cannot find the best. We cannot find the exact hit anymore, but we still go back to this group, but we can recover. And we can look at all the alignments that we find to evaluate them. Are these good alignments, or these kind of bad alignments, and so on. And we can do this for all of them. Here we have one that does where we were sadly not able to find anything. It might be for many reasons why we were not able to find anything. Maybe the best sequences in the database were just collapsed into a cluster, and then the representative was not similar enough, so nothing could be found. And for the other 17, we also mostly find like some good results. And here let's look at one more. Oops. Let's look at what we find in the PFAM. Another protein that we were successfully able to recover from the same order. Okay. So I'm ending this talk on a not very happy note. You can look up the special case where we use data from in this paper. The patient, even though they were very fast able to identify what the pathogen was, very sadly just not really any treatment for Povassan. So the doctors did the best they could with the patient's care, and eventually the patient was able to leave the facility, and there was some neurological recovery, but well, in this case there was just nothing really anyone could have done after the fact anymore. Okay, so, to not be too much a downer, but still, you can look up all the tools that we used here, and this really nice paper with a nice data set. And thanks to the well, all our funders. This work was only possible because we received like a lot of European money and other grants and so on. And Martin's group, well, you can do your sales pitch a bit.
Okay. So I just started a group here in March, and as already have 5 students, which is amazing, and we are hiring more postdocs and PhDs. So just if you're interested in metagenomics and this kind of work, if you like MM6 and you want to contribute something to it, then yeah, just write me in Twitter or email or whatever, and stay in contact.
Also, Seoul is an amazing city. You will get to live in Seoul.
Yeah. Okay.
Do we have anything more? Oh yeah.
This is more or less at the end of our part. We will still be around in the chat room, and also yeah, we could keep this open as well. For me, it doesn't make a difference if we close it or not. If somebody wants to ask questions, we can still answer them here in this forum. And we have still like 35 minutes to go.
You're muted, Silas. Hey, Silas?
Otherwise, you can have a look at yeah, both tutorials, continue with them, with the MM6 or with the metagenome-atlas. And we are happy that you joined our tutorial in quite a large number. And yeah, we hope you get in touch either by mail, Twitter, GitHub issues or whatever, or the chat we have prepared for you. And see you at the conference.
Bye, everyone. Okay, I can answer some questions still. So GTDB, it's not quite well built in yet. Am I still sharing? Yeah. It's not quite built in yet. There's some work to be done, but we have GTDB contributed, a script contributed for GTDB, how to convert taxonomy into something that MM6 can read. We will eventually work on this and improve it so it is integrated cleanly into this databases tool, but you could you can look into it. We haven't done much to evaluate it yet. And we are also working on more, for taxonomy, we are working also on other things. We are also working on a method that is not only good for annotating single reads, but a method that takes all our read annotation, all our read classifications and gives you the best possible, most specific label for a whole contig. Stay tuned, the preprints should be posted up soon for that. And I hope we can also keep on working and making MM6 better for taxonomy in, yeah, with all this work. Does the other question? I don't see the other question anymore. I don't know where it went. Okay.
I don't completely understand the question. It's when the reads were aligned back to the polyprotein sequence, were these all non-human reads available or after binning?
Oh, I don't see it anymore. But these are all only, we explicitly extracted only these 1,200 reads that MM6 was able to annotate as a pathogen, as a povasan. And only compared the povasan reference protein against these contigs that we assembled. I hope that helps with the question. Ah, wait, now I see more questions. Ah, how can I do nucleotide to nucleotide searches? Okay, so where was I? We can, we have many different search modes. There's this, let's just replace this.
One of my search type.
Search or easy search. Oops. We have many different modes here. One of them that you find is here, search type, and you can tell it what exactly you want. This is usually auto-detected, but in some cases where it's ambiguous, if you want a basically a T+X search or a T+ or a BLAST-N search or some other kind of variation, you have to specify exactly what you want to do with the nucleotide searches. MS6 has some limitations for nucleotide searches. It still does, it uses a banded alignment library, so it does not produce like classical full length alignment. But it is still a fast useful tool for nucleotide searches, I hope. With the gaps where in read. It's hard, yeah, it's difficult to say what exactly was going on. Oops, what's happening now? Ah, here. If you compare it with, I didn't prepare anything to compare it directly with each other, but you see that, oops, you see that these are similar, these are similar regions where we just have no reads at all, where there could not be anything assembled, and then there's also just reads that have such a huge variation that we just could not. Produce anything. Oops, where we could just not. Sorry, this Jupyter notebook is currently acting weird. Yes, you can produce. You can produce SAM output with. Where is it? 1.
Not 2. 1. Yeah.
Okay. Oops. And then you get the SAM output format. Can I get SAM? The recommendations how to set thresholds for searching against annotation databases. Well, it very much depends on what you search against and what you want to achieve. Like the default e-value thresholds, we just assume everything that you hit inside the e-value cutoff is homologous, and that's usually a very good assumption. If you want something more specific, you can restrict. You have various parameters to restrict MM6 on what alignments it produces. You can demand like a minimum length of alignment length. You can demand these coverage modes and so on to have more specific domain to search, for example, for a more specific domain structure or a full length thing. It's a very use case dependent question, I guess.
There's this publication by Yannick Malik from Jana Bromberg's lab. What they did is for annotation, they were interested in enzyme annotation and figure out if these enzymes are still functionally similar to each other. And they combined MM6 with profiles, so they actually built profiles. But then their criteria, so that they really can find really far homologous hit. But their main criteria is dependent on the alignment coverage and the sequence identity at that point. So the more coverage you have, the lower your sequence identity can be. So this is more or less a guide. So this is really like a field where you can do a lot of research in it. But that paper is already a good starting point, maybe to have a fast and reliable annotator for enzymes, for example.
Okay. Do you want to build it, Martin? Are there any plans to integrate the RefSeq contaminated clean genomes integrated into the MMSX default database. Good idea. We could.
Yeah. Yeah. We probably would need to run this contamination scan with every new release of GenBank to check for the new contaminations. But it would be fairly easy to also, if you want to do that yourself, it's easy to just use that list and just remove all of the identifiers that were marked as contamination. But yeah, that would be a good idea. We should talk about that.
Right now, we didn't even produce a default downloader for RefSeq because it's kind of complicated to download the RefSeq. It's split into so many parts. Yeah, but it's a good idea. We can do it. Okay, I think we can close the session. Thank you for listening. I encourage you to try out our Jupyter notebooks yourself and ask questions. I hope this was an interesting and fun experience to learn about these cases. I thank you all a lot for joining and taking part in this tutorial. Hope to see you all in person sometime.
Thank you.
Goodbye.